Although the quantitative proteomics, the qualitative proteomics, determination, the mass of the protein molecule, and they are very important in the study of the protein in biomolecules. But another important thing to study is the protein interactions, that how proteins, they interact with each other and what is their importance. This is what we will discuss in this module, that is the protein interaction. Now, the most proteins, they do not function in isolation. One must understand this, that there may be protein present, but it shall not act in isolation. But these protein molecules, they collaborate with each proteins. They collaborate, they associate with different proteins by participating in biochemical or developmental pathways. There are biochemical pathways, there are developmental pathways, and these different proteins, they participate in those pathways to function properly for a microorganism, for an organism in that sense. For example, let's study in the signal transduction pathways, the proteins. There are different proteins, they interact with each other so that the signal transduction is carried out. Many proteins form large multi-protein complexes. So there are large number of multi-protein complexes. Means that different proteins, they make complexes together, and that multi-protein complexes, they are dedicated to a specific task. That is, they are dedicated for a specific task, and that is such as ribosomes, which is a protein synthesis, or the proteasome, that is the protein degradation. That is where protein synthesis is required by proteasome. The different large multi-protein complexes, they will be formed and dedicated to this job. Similarly, different proteasomes for the protein degradation, a multi-degradation protein complex will be formed. Now, interaction of protein, that can give important clues about the function of newly discovered protein. You see, when they interact with each other, we can get different clues for getting the function of newly discovered protein which are present there. Now, just as example, let's study the yeast-2 hybrid analysis. That is, traditionally, protein-protein interactions have been detected by yeast-2 hybrid analysis and some proteome-wide studies of protein. That is, the proteome-wide studies, proteome studies of the yeast hybrid analysis was carried out. And these studies, that the protein interactions that have been performed using this technique, the technique is yeast-2 hybrid analysis, where the hybrid analysis was carried out. And again, we get the protein mass spectrometry to determine the mass of the different component of the peptides, and the investigators, they have used another ultra-sensitive protein mass spectrometry to do a better job of detecting protein-protein interaction. So to determine the protein-protein interaction, you need again a protein mass spectrometry and ultra-sensitive devices to determine that. Now, in that case, generating the tagged bait protein. So what we do first, we generate a tagged bait protein, which is shown there, and a yeast gene encoding a bait protein is engineered. So what happens? A bait protein is engineered and it includes the coding region for the tag. That is, that protein has got a coding region for a tag because it is tagged. Then isolating the complexes with the bait protein. That is, those bait proteins, they are isolated by which method? By immunoaffinity chromatography method. So what happens? That a yeast gene encoding bait protein is engineered to include the coding region, and then that bait protein is isolated, and the complexes which are isolated, they are analyzed by a immunoaffinity chromatography. With the immunoaffinity chromatography, it purifies and identifies the different proteins. By STPase is used to separate and purify the protein in the complex, as we have studied or described earlier. Now the whole procedure is shown in this diagram, where you see on the top there is an engineered bait protein. A tag is attached to this, then it is further degraded and isolated, again separated by affinity chromatography, and the different peptides, they are run through the SDS page. It gives us the different peptides where the further analysis is carried out precisely to determine this. So in summary, what we can see, that the several techniques are available to probe these protein-protein interaction. There are different techniques, as we have discussed earlier. Traditionally, what happened, that the yeast-2 hybrid analysis has been done, but now other methods, they are also available in addition to the yeast-2 hybrid method. And those methods include protein microarray, immunoaffinity chromatography, followed by the mass spectrometry, and the combination of these all experimental methods which are there. Such as another, a phage display with the computational methods, they are also available to perform this thing. One of the most useful fruits of such analysis is the discovery of the function for new protein. That is, for the protein interaction. And all these things, the important thing is, which comes out of this study, is the discovery of the function of new proteins which are present there, which are interacting in the pathways and will advancing or shall advance the knowledge of the proteomics.